Okay, Assalamu alaikum. Now we are beginning with our chapter number 3. Technically, this is our chapter 4 because we did cell membranes beforehand. And now this is chapter 3 in your CI syllabus, enzymes. It's an easy chapter, but obviously now you're in A level, so there are slightly more technical aspects that we need to cover for this chapter. So starting off with the properties of enzymes, which is that now we already knew about the fact that enzymes are protein in nature, right? But now we need to write that this is a globular protein. Okay? This is not a fibrous protein, this is a globular protein. Fibrous proteins have fibers and they're particularly responsible to provide structural supports like collagen. Okay? So enzyme, antibodies, and hemoglobin. All three of them are the example of globular proteins. Then we need them in small amounts. It has a tertiary structure because now we are done with biological molecules. We now know that all the proteins are tertiary in nature, but few of them are quaternary in structure. Quaternary are those which have more than 2 polypeptide chains. Okay? But before that, all the proteins are tertiary in structure. Then we are already aware about the fact that it is highly specific in nature in terms of pH and temperature, that it works at certain pH and it works in certain temperatures. Then this is a biological catalyst which speeds up the rate of reaction. And we have a substrate, we have an active site. So basically enzyme has a particular active site to which the complementary shape of substrate binds in order to form enzyme substrate complex, and then we can get our desired products. So it can catalyze metabolic reactions. Metabolic reactions are two in number: anabolic and catabolic. So basically enzymes are responsible in catalyzing anabolism and are responsible in catalyzing catabolic reactions as well. Okay? So anabolic reactions are those which are responsible to make something, while catabolism reactions are those, or catabolic reactions are those which are responsible to break down any substance. Okay? So anabolic reactions are synthesizing one, catabolic reactions are breaking down one. Then it lowers the activation energy. So basically when we write this line that enzymes speed up the rate of reaction. So now we need to write it, how can they speed up the rate of reaction? So basically they can speed up the rate of reaction by lowering the activation energy and by providing an alternate route for the reaction to take place. Okay, so it's not written over here that it provides an alternate route for the reaction to take place, but if you're making notes, then try to write this as well. Okay, one of the reasons is this one, that it lowers the activation energy. Other one is that it provides alternate pathway. Then enzymes are water soluble. They remain unchanged at the end of the reaction, and they could be intracellular and they could be extracellular. So basically when the question says, because this is a very repeated question and often comes in your papers, that if they are suggesting that one of the enzymes is particularly working inside the cell, then what is the category of that enzyme? So you'll write intracellular, that it's working inside the cell. And if they're saying that certain enzyme is working outside the cell, then you'll write extracellular. Okay? So this is very important. It will come when we'll practice the past papers. So these are all the properties, all the characteristics of enzymes. And if a question comes, then you can write about any one of them. Okay? Hey, we have working of enzymes. So I already mentioned that enzymes catalyze reactions, catalyze metabolic reactions. Metabolic reactions are anabolic and catabolic. Anabolic reactions are bond forming. Catabolic reactions are bond breaking. Then for an example that you can remember, that anabolic reactions are bond forming, and the example for anabolic reactions is photosynthesis. And you should also know about this fact that we need to hold the substrate at a particular angle and orientation so that a successful interaction between substrate and enzyme can take place and enzyme substrate complex can be made. Okay, then we have catabolic reactions, which could be breaking of lipids into fatty acid and glycerol, or breaking of proteins into amino acids, or breaking of cellulose into beta glucose. These are all the examples of catabolic reactions, and that chemical bonds are strained within the substrate until they break. So this is like a overview of what we have already studied in biological molecules. So you need to remember that enzymes work in two ways. Either they form a bond or they break a bond. You should remember the example for bond formation. You should remember the example for bond breaking. Then we also know that enzymes are specific in nature. So there are two types of specificities when we'll talk about enzymes. One of them is speckle fit, and other one is chemical fit. Because a speckle fit is, you are already aware of this, but you do not know this terminology. So this is an additional information that speckle fit is that complementary shape will fit into active site. Complementary shape of substrate, right? So substrate is the molecule which has to bind with the active site, and both of them should be specific in nature. They should be complementary in nature. Okay. So if they'll be complementary, then we can say that this is a speckle fit. Then chemical fit is that enzyme substrate must have complementary charges such as positive and negative. So for example, if your active site is negative, then your substrate should be positive, right? Because opposite charges attract each other while like charges repel each other. Okay, so we need to be mindful that we have speckle fit and we have chemical fit, and enzymes work in two ways. Either they form a bond or they break a bond. Then we are already aware of the lock and key hypothesis, right? You already did this in your O levels. The new part is induced fit. So lock and key hypothesis, a brief overview of this is that if you consider enzyme as a lock and you consider substrate as the key, and when lock binds with the key, and they should be complementary in shape, then your substrate binds to active site and non-covalent bonds are formed. So you need to now remember all the bonds that are forming between two of the molecules. Okay? So you can easily explain this. But now you need to understand induced fit mechanism. Okay? Because this is what you'll be tested on. You are not much tested on lock and key mechanism. You are now tested on induced fit mechanism. So it says these are basically two theories of the mechanism of enzymes. And this says that initial shape of active site may not be complementary to that of substrate, but as substrate enters active site, it induces conformational change in the shape of the enzyme that it perfectly fits into active site. So I know this is a lot of English that you need, it is a lot of English to take in. So what you can do is you can understand this example, and then you'll understand the entire mechanism of induced fit. That when we buy a new shoe, a new what do we say to those shoes that are for sporty purposes? Those shoes are very tight initially, right? When we put in our foot into those shoes that are completely brand new, so it's a little bit of tightness can be felt, right? But as you wear it repeatedly, then you'll feel that tightness of the shoe has been disappeared. Okay, so this is exactly the same mechanism for enzyme substrate, which we call induced fit, that they say that initially active site is not complementary to the shape of substrate, but with their interactions, the substrate induces a conformational change in the active site, and then it becomes perfectly fit. Okay? So is there any confusion in induced fit mechanism?
No, miss.
Okay. Achha ji. So this is a diagram of lock and key hypothesis that you can see that these are absolutely specific, or these are absolutely complementary active sites with the substrate. So this is a substrate molecule of sucrose. It binded with its active site. Then what happened is that two products, fructose and glucose, are obtained with the reaction of substrate sucrose with active site. Okay? So you need to remember that substrate enters the active site of enzyme, and then After that, you need to specifically write that enzyme substrate complex is made. This is made when substrates are binded with the active site. So you need to remember this word, enzyme substrate complex. Then afterwards, when the reaction has taken place and products are now being formed, then you need to mention that enzyme products complex is formed, and then you need to mention which products have been formed after this particular reaction. So this is the lock and key. Now this is the induced fit. Induced fit, like I said, that initially the active sites are not complementary. So after the interaction of substrate with the enzyme substrate complex is made with a certain conformational change that is induced among it, and then you can see an enzyme product complex is made, and then products are released. Okay, so far are we good? समझ आ रही है?
Yes.
All right, so now let's do measuring rate of reaction. So every time I teach this topic of rate of reactions, be it in biology or chemistry, I say that whenever we are talking about rate, you need to get this information immediately in your brain that now we are talking about time. Okay, rate means time. So how can we measure the rate of enzyme reaction is with substrate used or product form, whatever is given in your paper. Okay, kitna substrate have we used and kitna product has been formed over a certain period of time. So let's say substrate used was, I believe 10 concentration 10 moles per dm cube something. Okay, so I'm using the concentration unit. 10 moles per dm cube is the substrate that has been utilized over a time of 20 seconds. So now we are going to divide it and we are going to give the rate of enzyme reaction. Other than this, if they have not provided you with the substrate that is consumed, you can also write the numerical value or the magnitude of how much product is formed. Okay, so this is also an important question, that factors affecting rate. It's temperature and pH. For temperature, you have three categories: before optimum, at optimum, and beyond optimum. So higher the temperature, higher is the kinetic energy. So you need to know about this complete information, and it's better if you write this one because I've extracted this directly from your marking scheme. So let's just stick here that as we'll increase the temperature, kinetic energy will also increase, and at optimum temperature, we are aware of the fact that rate of reaction is highest due to greater number of collision, and collision is resulting from higher kinetic energy. Okay? So if you increase the temperature, you'll increase the kinetic energy, which will result in greater number of collisions. And then if we'll proceed beyond optimum temperature of a particular enzyme, then its hydrogen and hydrophobic bonds will be broken, because we initially studied that enzymes are tertiary in structure, right? So and we are also aware that in secondary structure, hydrogen bonds are made. Then we have hydrophobic bonds when your tertiary structure is being made. So if we'll go beyond optimum temperature, then hydrogen and hydrophobic bonds are broken and you lose your secondary structure, which will cause the loss of active site and which will result in deformation of enzymes. Okay? So it's important to mention, this is your keyword, secondary structure is lost. Why is it lost? Because of the loss of hydrogen and hydrophobic bonds, which will result in deformation of enzymes. Then we have pH. So pH is basically the measure of hydrogen ions in solution, and that enzymes work best at specific optimum pH, and any pH other than that will decrease the rate of reaction. Ionic and hydrogen bonds are disrupted, causing enzymes to be denatured. For pH, you need to remember that ionic and hydrogen bonds are disrupted. For temperature, you need to remember hydrogen and hydrophobic bonds are disturbed, which will deform your enzyme. Abdul Rahman, I saw you raising hand. Is everything okay?
Yes, I just wanted to know कि आपकी notes शेयर करेंगी?
जी notes to share होते हैं. Do you have the access of Drive? I shared the notes on Drive and on your WhatsApp group.
आप कर दें drive share.
Drive share with me. You need to search it. Usman, can you do it? Because I'm not sure where exactly, when exactly did I share the link of Drive.
ओके मिस, मैं फॉरवर्ड कर दूंगा क्लास के बाद.
हाँ जी, प्लीज
Okay,
याद से कर दीजिएगा. Thank you.
Thank you.
All right, so now we have limiting factor. So limiting factors are two: enzyme concentration and substrate concentration. At low enzyme concentration will become limiting factor. So limiting factor is basically any factor which is in a limited quantity. Okay, so anything which will be in a limited quantity will be the limiting factor of that particular reaction. Okay, if concentration of substrate is higher, then all enzymes will be saturated. Hence, enzymes will become the limiting factor, and which means that supply of enzyme will short. If I'll explain this to you in a mathematical format, then you'll be able to understand this more efficiently. For example, there are just 5 enzymes, okay, in a particular reaction, while you have 10 substrates that needs to be catalyzed. Okay, you have 10 substrates and you have 5 enzymes. This is not a very good example, but just for you to understand this amount of English, I'm explaining it in a mathematical format that you have 5 enzymes and you have 10 substrates that needs to be catalyzed. So basically होगा ये कि 5 enzymes will eventually they'll bind with only 10 substrates, right? And your how many are these? These are 4. So basically these are 5 enzymes, ठीक है? And 5 enzymes के साथ आपके 5 substrates ही bind हो सकते हैं, right? So 5 substrates binded with your 5 enzymes, and this means that now the remaining 5 substrates are now left alone. Okay, these are your 5 other substrates which are left alone and they do not have any enzyme until and unless a particular reaction takes place and then the substrate leaves and then substrate can bind. So basically, I'm just trying to say that enzyme here is in a limited quantity, which is reducing the rate of reaction. Okay, are you able to understand?
Yes.
Are you able to understand that if you increase the concentration of substrate, let's say there are 5 extra substrates over here, right? We do not have 5 more enzymes. So which means that 5 enzymes that are already present in this reaction, they are all now saturated. Saturated means that there is no more capacity for other substrates to be entertained, right? Make sense?
Yes.
Okay. All right. So this is now a very, very important thing that you need to understand. And this is something which is a part of your papers in all the years. Okay? Velocity max and Michaelis Menten constant Km values. So basically, let's just not go in this much amount of detail that's given over here. I'll explain you from this graph. Okay. So this is a very straight line. Acha, I'm drawing another graph. Okay, this is a good picture. This picture is of your book as well. Okay. So let's say this is your rate of reaction. Okay. This is exactly how you make graphs for any rate of reactions. All right. अब यहाँ पर you can see कि ये जो आपका graph है वो यहाँ पर आके कुछ terminate हो रहा है. It's terminating over here. So the maximum range where your enzyme could show the rate of reaction, this is your Vmax. Okay, Vmax is basically velocity max, which shows that this is the best working of enzyme, and this is like the maximal type, and this is the time where enzymes are completely bound to the substrate, and this is like the best efficiency of your enzymes, which is velocity max. Okay, if this is also my reaction, I need to see where is my reaction terminating, and I'll draw a dotted line from there, and the maximum value which I can get is of Vmax. Vmax shows that now at this point the enzymes and substrates are bounded with each other, and this is like the velocity maximum of your reaction. Have you understood velocity max?
मिस लेकिन ये भी तो हो सकता है कि सबस्ट्रेट ही हमारा लाइक सारा यूज हो गया हो
हाँ,
एंजाइम्स
तो
नहीं।
यहाँ पर इसीलिए तो आपका रिएक्शन पे टर्मिनेट हो रहा है ना.
हम्म।
This is like the maximum of the rate of reaction. यहाँ पे आपका सारा सबस्ट्रेट एंड एंजाइम एक दूसरे से बाउंड हुआ हुआ है. अब अगर कोई सैचुरेशन वाली बात है तो वो यहाँ रिकॉर्ड नहीं हो सकती ना, वो तो फिर स्ट्रेट लाइन आपकी हो जाएगी ऐसे. तो वही हमने उसी लाइन को तो पिक किया है. that this is the maximum. So with try to understand the word maximum. कि maximum means कि यहाँ पे rate of reaction आपका terminate होना शुरू हो गया या halt होना शुरू हो गया, right? Understandable? ठीक है. So now let's, acha, so this question comes in correlation with Km value, which is Michaelis Menten constant Km. If they'll give you Vmax, they'll for sure ask you Km because this is a slightly mathematical part. Acha, I'll explain this again with the mathematical value. Just give me a minute. This is my Vmax, ठीक है? अब let's say my Vmax is, if I make a dotted line, this is like 10, ठीक है? This is my Vmax, which is 10, whatever the unit is, meter per seconds. And acha, so if now I'm supposed to find out Km value. So Km value is basically the half of my velocity max. So what exactly is the half of 10?
5.
5. So I'll see where 5 is in the graph. I'll mark where 5 is. Then after marking 5, I'll see कि यहाँ पे reaction जो है वो कितने नीचे substrate concentration पर it is occurring. Whatever the concentration here is, अगर let's say यहाँ पे आपको दी हुई है 5, तो this is my Km value. How we are supposed to find Km value? We take a half of Vmax and half of Vmax पे फिर हम जाके नीचे substrate concentration को mark करते हैं. And after that, you're supposed to write that higher the Km value, lower will be the affinity of enzyme and substrate or vice versa. If your Km value is a lot, यहाँ पर तो देखें your Km value is very small ना, but अगर let's say आपकी Km value यहाँ पर कहीं आ रही हो, so it means that this is a very high Km value, and Km value depicts or it indicates how much enzyme affinity is there. And if there's a lot of Km value, then there is less affinity of enzyme and substrate. Less affinity means that some enzyme and substrate are going to bind with each other. Less enzyme substrate will bind with each other. And the lower the Km value, the higher the affinity of enzyme and substrate. ठीक है? If the Km value is lower, that means that maximum number of enzymes and substrates are binded with each other. Okay? So Vmax is the maximum reaction. And then when we take a half of Vmax, we need to find out the Km value from half Vmax. We go to the x-axis and look at the substrate concentration, and then you can easily write it in your answers. And then they'll ask you that how is the affinity, how can you determine the affinity? So if you'll see that there's a large value of Km, then you'll write that the higher the Km value, the lesser the affinity of enzyme and substrate is seen. Okay, make sense?
Yes.
All right. So now let's move to inhibitors. Inhibitors is also a new part for you because you have not studied in your O levels. So inhibitors are those which basically try to halt the reaction, or basically they try to stop the reaction by any means. Okay, so basically we have two types of inhibitors, competitive and non-competitive. So competitive inhibitors are very easy to understand. If I'll show you the diagram, it will be much easier for you to understand. See. So inhibitors are responsible to halt the reactions, okay? And now with this, you can understand the fact that substrate and inhibitor are of same shape. So if inhibitor binds with the enzyme, it will easily bind. Why? Because they have similar, or they have specific, or they have complementary shapes. So if inhibitor will bind with enzyme, then your rate of reaction will decrease. And why will it decrease? Because inhibitor has occupied the active site of enzyme. Make sense?
Yes.
Okay, so what exactly will happen? That your rate of reaction will decrease, but this is a reversible process. How is this a reversible process? If we'll increase the concentration of substrate, then there will be more chance for substrate to bind with the enzyme rather than inhibitor binding with your enzyme. Okay? So if we increase the substrate, then this will become a reversible process. And what exactly a competitive inhibitor is, that a similar shape of competitive inhibitor like that of substrate binds to the active site of enzyme and the final product is not made. Because obviously how a product will be made. It's an inhibitor. It is there to stop the reaction. It is not there to catalyze a reaction. Catalysis of the reaction is responsibility of substrate, right? So if substrate will not bind, then your reaction will not take place, or your rate of reaction will decrease. While a non-competitive inhibitor is a inhibitor which is not complementary to the shape of active site. Rather, it binds to any site of the enzyme, and any other site of enzyme other than active site is known as allosteric site. Okay? It often comes in your paper that any site other than active site of enzyme is known as allosteric site. So basically, in a non-competitive inhibitor, it basically binds with the allosteric site of enzyme, and it results in the distortion of enzymes. Okay? So this is how a non-competitive inhibition takes place. Okay, this is your enzyme, this is your proper active site, this is the substrate, but the non-competitive inhibitor binded with enzyme in such a way that it's not binded on the active site, it is binded on a allosteric site. Okay, so if it will bind with allosteric site, it will distort the shape of the enzyme, and your enzyme will eventually deform, or your enzyme will eventually disrupt. And if it will disrupt, then it cannot be reversed, right? Because once you lose the shape of enzyme, you cannot reverse it. So this is a non-reversible process, because you lose the shape of enzyme if non-competitive inhibitor binds with allosteric site. Make sense?
Yes.
Okay. All right. So the last subtopic, basically this is the second last subtopic of enzymes, which is feedback inhibition. So you see that there are many enzymes involved, okay? And you see that if there are enzymes, then for sure you'll have products. Because koi na koi reaction is being catalyzed at that time. So you can see that at the last you can see product 3 is your end product. Let's say that your level of product 3 by the end of the reaction is increasing and increasing. So if it will increase to a dangerously high level, what will exactly happen? An inhibition of enzyme will take place. And how is that inhibition going to take place? That inhibition is going to take place by acting upon enzyme number 1. Enzyme number 1, why is it acting on enzyme number 1? Any guesses? If product 3 is increasing, then inhibition will take place on enzyme 1 to reduce the concentration of product 1, and eventually less products of product 2 will be made, and eventually less products of product 3 will be made. Okay? So eventually the levels of products will decrease to again a very dangerously low level, and then again your inhibition will take place, and your enzyme 1 will increase the activity in order to produce more product 1, then more product 2, and then more product 3. And if it will increase to a dangerously high level, then inhibition will take place again on enzyme 1. So this comes in your P1 and in your P2s as well, that agar product 3 जो है वो बढ़ रही है, so which enzyme should be inhibited. So बच्चे often write enzyme 3. It's not enzyme 3, because enzyme 3 से तो सिर्फ product 3 की आपने concentration कम करी, आपको पूरे reaction की concentration you were supposed to lower it. And how are you supposed to lower it? By inhibiting enzyme 1. Enzyme 1 will inhibit, then product 1 will decrease, product 2 will decrease, product 3 will eventually decrease. जब product 3 बहुत ज़्यादा decrease हो जाएगी, again enzyme 1 पे जो है वो inhibition होगी, and then enzyme 1 product 1 को बढ़ाएगा, product 2 को बढ़ाएगा, and product 3 को बढ़ाएगा. Make sense? So this end product inhibition finally controls the levels of product 3 between narrow upper and lower limits, and this is an example of feedback mechanism. Make sense?
Yes. वैसे हम इस प्रोसेस को